My name is Kity Požek, and I am now a PhD student at the Faculty of Medicine in Ljubljana, Slovenia. I am working as a young researcher at the Jozef Stefan Institute under supervision of Professor Dr. Igor Križaj and Assistant Professor Dr. Adriana Leonardi. So I will present our work that offers a new insight into the origins of snake venom Disintegrin-Like Cysteine-Rich proteins, or DC proteins. And this work was published last year in the journal Toxins. So I invite you all to read the article for more details on what I will present today. The first genomic part was the work of our whole group, while the second part was the work for my master thesis. So DC proteins are snake venom toxins that consist of two non-catalytic domains, the Disintegrin-Like domain and the Cysteine-Rich domain. Most of them affect hemostasis or are involved in the inflammatory response to snake bite and venoming. And the exact origin of DC proteins is still controversial, although it is clearly associated with snake venom metalloproteinases, or SVMPs. SVMPs are among the most abundant toxins in viperine venoms, and they are categorized into three classes according to their domain structure and into subclasses according to their post-translational processing. Evolutionarily, the oldest among SVMPs is the P-IIIe class, and it evolved likely from the Adam proteins. SVMPs are typically affect hemostasis and relevant tissues and cause hemorrhage. DC proteins have long been regarded just as products of post-translational processing of P3 SVMPs, as it was shown in vitro that proteolysis or autolysis of these SVMPs produce DC fragments. However, this is unlikely to happen in the acidic environment of the venom gland, and moreover, the DC fragments produced by autolysis of, for example, jararagin, are different in their structure from jararagin C, which is a DC protein which was isolated from the venom. So the origin of some DC proteins could not be explained by autolysis. In the cDNA library of the most venomous European snake, the nosehorn viper, we found a transcript of a gene that has a typical SVMP pre-pro domain and a truncated Disintegrin-Like domain and a Cysteine-Rich domain. So it lacks the entire Metalloproteinase domain and part of the Disintegrin-Like domain. It was named VAMP33, and it was also detected at the protein level in the nosehorn viper venom proteome. We hypothesized that VAMP33 originates from its own gene, so it is transcribed translated from a DC gene, which evolved from the ancestral P3 snake venom metalloproteinases and now represents a new P3E subclass of SVMPs. We screened the first draft of the nosehorn viper genome with the coding sequence of the protein precursor of VAMP33, and we found its gene and determined its gene structure. And we compared it to the gene structure of another SVMP, VAMP32, which is a typical 17-exon gene coding for a P3D subclass SVMP, which was also detected at the protein level. As you can see, they have similar length of the matching exons and similar positions of introns, as well as a very high similarity in their protein region, primary structure, which all indicates their common origin. So we concluded that VAMP33 is in fact transcribed and translated from its own gene, which evolved from the ancestral P3 SVMP by gene duplication and subsequent domain loss, and is now classified in the new P3E subclass of SVMPs. So the second part of my talk will be about the protein VAMP33, so the first member of this new P3E subclass of SVMPs. This is a 21 kilodalton glycoprotein, which has 17 cysteine residues, meaning that one of them must be unpaired, and thus it has a free thiol group. Proteins with similar domain structure are inhibitors of platelet aggregation and contribute to the anticoagulant effect of the venom. So our aim was to isolate VAA-MP3/3 from the venom and to elucidate its biochemical properties as well as its role in the venom, especially its effect on blood. So to predict some of its key structural properties, we prepared a 3D model by homology modeling. We used the crystal structure of Agistrodon acutus hemorrhagin 4 as a template. And as you can see, we found that VAA-MP3/3 has a potential N-glycosylation site on its surface and an S-ECD motif in its disintegrin-like domain. This is a highly conserved motif among DC proteins, and it is known to mediate their antithrombotic activity. Another potential site of interaction with its biological targets is a hypervariable region, which is located in its cysteine-rich domain. So we developed an efficient isolation procedure to isolate VAA-MP3/3 from crude snake venom, and we found that it consists about 0.8 mass% of the nose-corn viper venom. This procedure included covalent chromatography, and binding of this toxin to thiolcephalose confirmed that it has a free cysteine on its surface. So we carboxymethylated this protein and in this way modified the cysteine residues so we could detect them by Edman sequencing. And we located the free cysteine at position 6 in most molecules, while 1 in 10 molecules had a free cysteine at position 19. And these are two states of the molecule, and by switching among them, there could be some difference in their biological function. So, as you can see, VAA-MP3/3 appears predominantly as a 21 kilodalton monomer, but can also form dimers of apparent 35 kilodaltons, and this could be covalent dimers formed between the free cysteines. So, using two-dimensional polyacrylamide gel electrophoresis, we found that we isolated a mixture of 6 isoforms with isoelectric points ranging between 4.5 and 5.1. The predominant isoform had the lowest piezoelectric point, and we could isolate 3 isoforms using reverse phase HPLC. All of them had the N-terminal sequence of RAGTE. We believe that these are glycoforms, as glycans can make proteins more acidic and hydrophilic. So we used PNGF to deglycosylate VAA-MP3/3, and we found that it has 4 kilodaltons of N-asparagine-linked glycans. So we used thermal shift assays to assess the suitability of buffers for storage, isolation, and functional testing of VAA-MP3/3, and we found that it aggregates in buffers with pH below 3 and is most stable in buffers with pH above 6. We could stabilize it with addition of calcium ions, but no other additives we tested could further stabilize it. So then we tested its effect on human platelets using turbidometry. And as predicted, it inhibited platelet aggregation induced by different agonists with similar potency to other disintegrin-like cysteine-rich proteins. The mechanism of its action remains to be investigated, but we believe that it acts similar to other DC proteins by binding of its SCCD motif or its hypervariable region to its to platelet receptors. We showed that it does not affect ristocetin-induced platelet agglutination, and nor did it affect blood coagulation times. So it acts specifically to inhibit platelet aggregation. And to conclude, we have confirmed that VAA-MP33 belongs to the new P-IIIe subclass of SVMPs, and we developed an efficient procedure for its isolation from crude nose-horn viper venom. We described some of its biochemical and functional properties, and we confirmed that it inhibits platelet aggregation. We are now preparing a recombinant protein for use in place of the natural one in further studies of its biological function and underlying mechanisms, which could lead towards the development of new antithrombotic drugs. So I would like to thank everyone who contributed to this research, especially my mentors and our colleagues who worked on the genomic part of the project. And of course, I thank you all for your attention.
Thank you very much, Kity. Super interesting topic. So, any questions from the audience? Yes, no, maybe. Then I will start. You showed that the DC genes are highly similar to the metalloproteinase original genes. Did you look into the genome where the DC genes are located? Because on the metalloproteinases, it's known that they are highly repetitive behind each other. Is it the same for the DC genes?
This gene was located about 400 base pairs from the one I showed, so the P3D SVMP, which was called VAMP32. So it is like close to the one of the P3 class.
So it's most likely like all the other evolutionary steps from the metalloproteinase that it has a duplication and then
the
truncation in the middle. And on one of your latest slides, you showed that the pH increase or decrease the activity. Is it possible after low pH, like 3, to go up to a higher pH, and is it then still reactive, or does it lose the activity completely?
So we only tested the stability. So this was the thermal shift assay, but we supposed, yes, if the structure is changed, then also the function should be compromised. But it is usually, we didn't test it, but it's usually not good recovery after this aggregation. But of course, it is not totally aggregated. There might be some of it recovered after changing the pH back to higher, more stable states.
So I think about that most of the reverse phase protocols use an acid running buffer, so that you have formic acid or something else in the acetonitrile, and there you're most often below 3 pH, so it would generate always an inactive DC protein if it's not reversible, right?
Yes, we used reverse phase HPLC only to separate the isoforms, just for the biochemical tests. But we then, when we were preparing the recombinant protein, we used the alkaline conditions for reverse phase HPLC. We used ammonium bicarbonate buffer. So I know it about pH 8. And we could prevent disaggregation and loss of the yield, lower yield, especially.
So I think that might be a really important point for other purifications and other venoms. So if you just make an HPLC purification of your peptide or your protein, then it might lose the activity because the system always runs on super low pH.
Yeah. We took this into consideration.
That's interesting. Any further questions? I saw a hand from Eric.
Yeah, you stole mine, so all good.
Sorry.
Thank you. No worries.
Okay. I don't see any further questions. Sorry?
I would like to add that this is not a catalytically active SVMP, and for the SVMPs that have metalloproteinase domain, it is very important not to just forget about this pH affected activity, because they really lose catalytic activity after this low pH isolation procedures.